Nous avons le plaisir en fait de recevoir le professeur Danovaro, qui est le récipiendaire du prix ENI Environnement de 2013. Donc je vais le présenter dans quelques minutes, vous raconter de façon très courte en fait pourquoi finalement il a eu ce prix, mais la meilleure présentation qu'on puisse faire, c'est la conférence qu'il va donner en fait dans quelques minutes. Mais avant de le présenter, le professeur Gérard Ferré de l'Académie des sciences voudrait un petit peu donner de l'information sur qu'est-ce que c'est que ce Tenny Award et vous commenter un petit peu en fait ce qu'il pense au sujet de cet award.
Merci Clément, bonsoir à tous. Oui, effectivement, je vais dire ce que je pense de cet Tenny Award. Bon, d'abord, historiquement, il a été créé en 2007. 2007, avec une ambition affichée sur le moyen terme, c'est de devenir l'équivalent du Nobel pour l'environnement et l'énergie. Celui qui a créé ce prix s'appelle Leonardo Baugieri et qui a quitté l'ENI pour l'instant. Mais l'idée est excellente et toute l'organisation de l'ENI a travaillé pour que justement ce prix devienne un des prix les plus fameux au monde, à côté du Nobel et du Japan Prize of Science. Donc il y a beaucoup de candidats. Les candidatures sont libres. Il y a beaucoup de candidats, on est entre 1000 et 1500 tous les ans. La sélection est féroce parce qu'il y a donc ce prix général est divisé en 4 catégories. 2 concernent le pétrole, puisque je vous rappelle que l'ENI, c'est une des 5 grandes compagnies pétrolières au monde. Donc, 4 catégories, 2 réservées au pétrole, la recherche amont et l'exploitation aval. Le 3e prix est sur les énergies renouvelables, parce qu'il ne faut jamais se contenter de l'existant, mais toujours avoir une projection vers l'avenir. Et le 4e, c'est l'environnement. Alors ça veut dire que le spectre à chaque fois est large, mais la sélection est féroce, se fait en plusieurs temps, et tout se décide avec un jury d'une trentaine de personnes. Alors ce jury est international, il y a des Américains avec des prix Nobel, des Américains, des Anglais, des Allemands, des collègues italiens, bien sûr, ça va de soi. Et c'est comme pour le Nobel. Il n'y a pas encore eu de mauvais Enni Award. Et c'est vraiment, j'en parle par expérience, ce prix, d'abord, est maintenant reconnu dans le monde entier. La preuve, 1500 candidatures, vous savez, ça ne s'invente pas. Ne récompense que des grands scientifiques. Ce n'est pas des prix de fin de carrière, je le précise. C'est des prix qui récompensent l'originalité et l'innovation. Donc voilà ce qu'est ce prix. Et cette année, c'est notre ami Roberto qui a été élu pratiquement à l'unanimité au premier tour. Et donc je ne vais pas amputer son temps de parole. Avant qu'il vous parle lui-même, je vais dire à Clément qu'il vous présente le lauréat de l'ENI Award 2013.
Bon, merci Gérard. Donc, je vais présenter donc Roberto Danovaro. Donc, en fait, Roberto Danovaro est professeur de classe exceptionnelle en biologie marine et écologie à l'Institut Polytechnic, donc l'université de Marche en Italie. Il est également directeur du département des sciences de la vie et des sciences environnementales. Donc, je vais passer assez vite sur la partie doctorale. Sachez qu'il a fait de nombreuses post-doc, beaucoup d'activités en post-doc, mais de façon assez large, je dirais dans des pays comme l'Irlande, la Belgique, la France. Donc, professeur Danovaro parle très très bien le français. Il va faire quand même son exposé en anglais parce que techniquement c'est toujours plus facile, mais par contre la discussion que j'ai eue avec lui, c'est qu'il répondra sans aucun problème aux questions en français ou en anglais selon votre bon désir. Donc en fait il a été nommé full professeur très tôt, à l'âge de 35 ans, et il s'est engagé dans de nombreux projets européens, une quinzaine, qui lui ont permis finalement de développer des nouveaux outils très originaux pour comprendre justement la biodiversité marine et tout ce qui est associé justement aux problèmes d'environnement associés à la biodiversité marine. Alors il a de nombreuses publications dans des journaux relativement de très haute qualité, Nature, Science par exemple. Et pour cet ensemble de travaux, indépendamment du prix ENI qu'il vient de recevoir, donc protection de l'environnement, centré également, vous voyez, de son exposé, ça va être le rôle et la synergie qu'il y a entre les océans, les microbes et la synergie associée finalement au changement climatique. Mais il avait déjà reçu, avant ce prix ENI, en fait, la médaille d'or du Prince de Monaco, qui a été donnée par la Société française d'océanographie, ça c'était en 2012, et The World Biology Prize également. Il est également listé parmi les plus illustres chercheurs italiens dans ce domaine, en particulier via l'Académie des sciences. Mais professeur Danovaro, indépendamment de cette très large activité, je dirais, scientifique, il a également une activité, un sens, je dirais, des activités collectives. Il a été président de la Société italienne d'écologie, puis président de la Société italienne de l'Association d'océanographie et de limnologie entre 2003 et 2006. Il a également été président de the European Federation of Marine Science and Technology Societies. Donc je ne voudrais pas être trop long. Le mieux, c'est que je lui donne la parole pour qu'il vous montre toutes les choses très intéressantes qu'il fait et qu'il a découvertes.
Merci. Merci à tous. Merci, Monsieur le Président, Professeur Sanchez. Merci à vous. Et merci pour l'invitation. C'est un honneur, un plaisir. C'est vraiment incroyable d'être ici. Donc, merci pour ça. As anticipated, I'm going to switch in English, but feel free of doing any question you want in any language, French, or English, or Spanish. In any case, the outline of my talk is trying to go from the big to the small, understanding whether our appreciation of the marine environment is generally based on what we see, but the most important things, or let's say, other important things that rule the oceans are very tiny, very microscopic, and they interact with the old systems as a guarantee for the global functioning of the biosphere. So that's is more or less the structure of the talk. So starting presenting some components of the life, especially highlighting some discoveries that were acquired the last 10 years during the Census of Marine Life, which was a very long and important project, and then going into the role of microbes, into the effects, not just climate change, but the effects of climate change in the oceans. And then that was the topic of the project that was receiving the award, how microbes can interact with climate change. The planet is blue, we know. The 70% of the planet is covered by oceans. Not all of us know that 90 to 95% of the volume of the biosphere is marine water, is made by oceanic waters. And so the largest majority of the space inhabited by life is made by the oceans. But given this huge amount of water, huge surface, we are talking about more than 374 million square kilometers. How much do we know? We have explored only 5% of the overall surface by remote tools, multi-beams, thermographic or topographic understanding. So very little is known. So when we have a topography of the bottom, when we describe the oceanic floor, we just guess what is in between different dots we took here and there. And if I have to evaluate how much we have really taken and investigated in detail, like if we go to the moon and we take a stone and we bring it back and we analyze it. The same is for the deep oceans. If we calculate the surface, that is about a couple of courts for football courts. So almost nothing compared to the huge dimension of the oceans. So life is not just on the surface. It's not just what we see from on our summer, on the vacation at sea, snorkeling with a mask, but most of it is hidden in the ocean interior, and in particular in the deep oceanic floor, so, which is quite complicated, highly complex from a topographic point of view. It's full of canyons, structures, seamounts elevating 4 kilometers from the seafloor and so on, and even smaller structures that I'll show you later on. The deep ocean interior always was, very intriguing for humans. I just suppose here Jules Verne, you know better than me, that was describing, was hypothesizing what was hidden in the ocean interior in the 20 milieux sous le mer. He hypothesized that was full of creatures. Some of them were even realistic, large animals, strange creatures, monsters, giant squids, and so on. It was missing a few points. It was hypothesizing strange algae. Well, there are strange algae, not maybe photosynthesizing in the deep, but because there is no light. But now we discovered that there are maybe forams carrying, we saw animals carrying photosynthetic systems in the deep, so strange things are happening. Giant calamars, and we know are there. Just a few years ago, we were able to take the first pictures. The Japanese colleagues of the JAMSTEC were able to take some pictures. And then animals passing through the Suez Channel, and then Antarctica. And that was a big mistake, because it was imagined that it was possible to pass beneath Antarctica. But now we know that is a continent that is clearly not possible. So which are the new frontiers of research? Is that the life of marine biologists is that easy? Well, depends on the people you find at sea. This is a white shark, and that is a scuba diver trying to explain that they just need a few images. He's still alive, and the shark too. But the results, some of the results I'm showing here are not just because of my work. It's because of a network that involved thousands of scientists, a huge amount of days at sea, a huge number of species discovered. That was one of the most amazing projects even conceived. It was ranked among the top 5 projects for humanity and was awarded by the Cosmos Prize. Which was the objective? The first homework that God gave to the humanity was giving a name to each species on Earth. And we still have completed the human genome, but still we don't know even the number of species inhabiting our Earth. That is not just a matter of curiosity. It's a matter of the huge number of products, enzymes, proteins, or whatever that we can extract and use also from these organisms. And most of them are hidden, as I was anticipating, in parts which are difficult to explore. And in certain parts, here we are 80-meter depth. You can also go by diving, but in most cases, this is not, even 80 meters, working at 80 meters depth is quite complicated. So in most cases, we need a sophisticated technology to work there and to understand processes. We were thinking that the deep ocean was a kind of desert with quite homogeneous, quite stable, with no changes. And now we discover that there are cascades of events, there are huge phenomena occurring, and it's highly dynamic. This is not a coral reef. as we intend to be a tropical coral reef. This is a coral reef occurring at 1,000 meter depth, so without zooxanthellae and symbionts that use light, but they create reefs as well, which are full of life, maybe every single reef hosts more than 500 species and a huge also biomasses. And that's why this is also a target for fishermen for going there. And this is one of the pictures that was taken by the project just a few months before the Deepwater Horizon in the Gulf of Mexico. So when the tragedy of the BP Deepwater Horizon occurred at 1,500 meter depth approximately, the life of that depth was that one. If we don't know that anything was there, we could not even imagine about the impacts that were due to this accident. And it's strange because, by the way, the Gulf of Mexico is very similar for many reasons to the Mediterranean Sea. It's a semi-enclosed system and has exactly the same number of species. 17,000 species there and 17,000 species in the Mediterranean. And then if we start exploring a little bit better the vastity of the oceans, we discover that there's a huge number of seamounts. So there are just mountains starting from the seafloor, but they do not reach the surface. They do not appear as islands. So if you don't pass over them, you don't see them. And the number is still a guess. So we made an estimate. Could be maybe 30,000, maybe 100,000. We were just exploring a part of the Cordillera Sísmico, going to the Easter Islands for an expedition, and we found 3 new seamounts there. So these are kind of strange islands because, as you will see, they host huge amount of species. Strange species like these, which are quite strange for our usual imagination of how a fish species is, because they don't need to move. They stay on the island. They just walk around the island. And most of the species, 50% of the species, 1 over 2, is endemic. You can find them only in that island, only in that system. So if you lose them forever in the world. Another thing is that we think about sharks. Jaws, the movie that was described with the shark with the fin, over the water, so you can see the sharks. And so we are afraid about sharks that we can see. But a large number of sharks is in the deep, and there are sharks occurring even at 4,000 meter depth in the deep. And they are playing an important role there. And we are just discovering there. But as soon as you put some food there, in a couple of hours, a big shark, a 6-gill shark like this, comes to visit you. And then the abyss. These strange creatures here are holoturians, sea cucumbers. They have increased, due to climate change likely, by 50,000 times in just 15 years. So what's going on there, we don't know exactly. But everything is linked. They reproduce thanks to the phytoplankton settling down from the surface, which is carrying carotenes that activate their reproductive system. So it's a kind of connection between different things. Even a creature inhabiting the abyss depends on the signals coming from the surface. And then we are also discovering that the abyss is dark, of course, but there is an amazing amount of bioluminescence there, which is going to be explored, which is even confounding physical experiments in kilometer 3 experiments in the Mediterranean Sea, out of Sicily. They were unable to detect some variables because of the huge amount of bioluminescence, as well as the communications of the whales that were passing through, the tooth whales were passing through, and so we're discovering by chance that there is really a lot of life. Say also controlling and contaminating life. For instance, we are used to see fish. This is a fish, Macropina microstoma, that they found they were unable to collect yet, but just to take some pictures in the movie. That is a frontal view. As you can see also the ocular globes there, which is quite unusual because we know the fish have a lateral view in this sense. So even the brain system has evolved in a special manner there. And then strange things like Dumbo octopus, for years this octopus was just recorded but never, catch up because they were unable to take it for further investigations. And all of these systems work thanks to association with symbiotic prokaryotes that allow them to have spatial powers. And then when we think that hydrocarbons are a kind of contamination and we are afraid of pollution to the hydrocarbon, we should understand that in the reality there are an enormous amount of ecosystems in the world that depends on the coal seeps and depends on the springs and the release of methane and also of heavy hydrocarbons, and there is enormous amount of life depending on these systems, not just microbial mats like this that can use methane, but also an entire food chain up to fish and some species like this, the Melibrachia lumesi. They host, they are kind of worms, like polychaetes, evolved in a certain manner, similar to. They have evolved in symbiotic relationships. They use methane. But the most important thing, these are single animals able to live for more than 600 years. So when we look for the secret of life and the generation of life, we do not understand which is the longest life you can live, and we make expectation for the human beings living up to under 50 years because of the degeneration of the DNA. Why not investigating the DNA of these components that they can live as a single, not as a clonal organism like a coral, but as a single organism can live for 600 years and maybe more? How did this solve the problem of the degeneration of the DNA during replication, for instance? So we have also tools for investigating spatial processes that we are unable to solve using humans as a model, for instance. And then the problem of the amount of asphalt. Even this structure is not the result of a sunk of a carrier of petrol or whatever accident, but it's just a natural emergence of asphalt that is inhabited by organisms that use these components thanks to associations. And this is not a grassland, but is full of animals like those described inhabiting the system. And these are bivalves that size. Half of their body, the soft body mass, is represented by microbes. They live in association with that. So we have systems which are very extreme. This is the Yeti crab, described in 2005, and reported also in the journal of Mickey here, you can see. So that means that it became very famous. And this crab was just described in some anthropologic remains from the ancient, manufacts of Polynesia, but never found alive because they live at 4,000 meter depth. So how the local population was able to describe the species is still a mystery. But in any case, life is possible beyond any possible, apparently impossible limit and constraint. The animals you can see here, Riftia pachyptila, you see the red pinnacle, their blood contains hemoglobin like humans because it has the highest affinity for oxygen, for instance. Or other components, for instance, the emergence of vents from the lava and the emissions from the seafloor can reach temperatures up to 400 degrees and more, recently has been discovered. And some organisms, and the name is very, very intriguing, Alvinella pompeiana from Pompeii, they can live up to under 50 degrees without. Now, under 50 degrees for some of you is familiar because it's close to the temperature in which we cook the cake in the oven, okay? And they are able to develop a system to cool down the temperature in association, again, with microbes and to resist that system and temperatures. And this is a beautiful paraprocess in which they use sulfuric acid and methane to grow the bacteria, and the bacteria release amino acids, and they live upon that release. And this is another discovery I made in the deep basin of the Mediterranean. In the most extreme system on Earth is a kind of salamoia. The salinity is not 35 per thousand, but 20%. No oxygen at all. And we found these animals and 3 new species for science that were the first multicellular animals able to live all their life cycle without oxygen. So we're not parasites living just a part of their life in absence of oxygen. They were the first animals. And there was an important outcomes of this, because we were, took 10 years to demonstrate that they were alive and they were not just cadavers. But then these very tiny animals, because they are maybe 1/10 of a millimeter, so under microns. But they are opening new perspectives also for how life can survive in absence of oxygen. At the end of this, we are more like Socrates, able to say more what we don't know that we do know, because the census of the life and the potential discoveries at the beginning. And for most components, we just know maybe, say, 50%, in some cases, 20% of it. And we don't know the majority of the species living there. We know a little bit better the large animals, but we certainly know less the small components. So let's move to the second part of the talk and dealing with the contribution of microbes to this. What about microbes? How many microbes are there? How are they important? Because when I started my PhD, I was looking for investigating the microbes. And my PhD supervisor said, well, I don't really think microbes exist in the oceans. So please, try to find out another topic. Now we know. It was a few years ago, but it was a little bit ignorant also, I have to say. Now we know that there are from 10 to 30 to 10 to 31 microbes just in the oceanic domain. And in just 1 kilogram of sediments, we can find 1,000 billion viruses. So this is a kind of image showing what is my view that was realized by the graphic designer, Michael Tangalini, of the surface of this deep sea floor is full of prokaryotes, bacteria, archaea, attacked by viruses in a perennial competition and fight. If you look at all components, large components, mega, big things, macro, large animals, meio, tiny animals, all of these decrease exponentially with increasing water depth. This is a logarithmic scale. Some others can even increase with increasing water depth, in particular, so prokaryotes, bacteria in particular, and viruses can increase in the overall abundance the deeper you go in the ocean. So we were thinking that the oceans were kind of deserts. Now we know that they are, full of life, Dumbo octopus, big sharks, and we discover that the largest majority of the biomass, and definitely the abundance, is made of microbes, in particular prokaryotes and viruses, which are, in overall, more abundant in the abyss than in the coastal areas. That means that if there are more there are more species, more genes, more opportunity for bioprospecting. So we have to go deeper to discover novel secrets. And if you go from the shallow water systems where animals, the green part, dominates, to the deep waters, you see there's a shift from large form of life in the surface. Then here we are in the Battial, 1,000, 3,000 meters. Here we are in the abyss. In the abyss, there is a full domination in terms of biomass of the microbes. And if we convert that in terms of paragrams of carbon per year as a production of biomass, we discover that a value close to 80, 90% of the overall carbon production, and I will say also nitrogen production in organic nitrogen production in the oceans is made by these tiny microbial components. So they are definitely important. They rule the world. They decompose organic matter. They guarantee the regeneration of the nutrients that sustain the primary production of the algae. But what happens really, as I anticipated, is a continuous perennial battle between microbes and their killers, the viruses, that continues to kill and destroy these components, releasing the DNA and recreating what is a kind of primeval, primordial soup, a kind of system in which in the saline medium you have a lot of life forms around the soup of amino acids, of dissolved DNA, extracellular DNA, a lot of genes which are released. Just to give you an idea, we think that DNA is part of life. It's like a soul. When we die, the DNA dies and is destroyed. It's not so. The DNA is a quite conservative molecule, and even the genes can be preserved. So even their functions can be preserved. We demonstrated that once released from the cells, the DNA of the animals and the DNA of the microbes can persist in the environment and can be used by other organisms acquiring the functions of these genes. So there is a kind of acquiring a superpower, extra functions. As a temporary form or as a permanent form, they can exchange genes, can exchange functions, and there are 9 times more DNA outside the cells than inside the cells. So if you quantify the total DNA, you find 10. Just 1 is within the cells, the rest is outside. So the important thing is that the genes running the systems are viruses. They kill everything, everywhere, always. As soon as there is a host, there is a virus killing it. They kill the prokaryotes in this case. They release this DNA. The DNA can be absorbed to particles or remain dissolved in the water and eventually used by the other prokaryotes either in form of genes, either in form of nutrients. Yes, because we also demonstrated, this is a paper we published on Science, that almost half of the nutrient requirement, especially in terms of phosphorus, because DNA is the richest molecule in terms of organic phosphorus we have, is used, and phosphorus is a very limiting compound for all life forms, is used by ingesting and digesting extracellular DNA. So at the end, we have to say that viral infections, like lions in the savannah, they kill the gazelle and so on, they are extremely important to stimulate the growth by killing some forms, giving some nutrients to others, and sustaining the production, different forms, especially in systems like in the deep, like in the largest part of the biospheres, which are limiting in terms of nitrogen and phosphorus for life. And after the microbes, another nice discovery is a paper just submitted to Nature Geoscience. Nice discovery we made is that the second largest portion of the biomass on Earth and in the oceans after the bacteria and the archaea are the viruses. The viruses are so important in numerical terms that even though they are very, very small, much smaller than prokaryotes, overall they are the second component in terms of biomass. If we can define virus, a life form, let's say, in terms of biomass, after microbes. So they decay, the decomposition contributes to a large extent even to the biogeochemical processes and to release of important components such as nitrogen and phosphorus. So at the end, one important concept is that life and death are tightly connected. We kill other components, release some components, and these components sustain life another time. So it's a cycling which goes in two directions. One is what we understood and we learned at school, you know, the trophic cycle, the food chain. But another one goes through as a trophic source, the lower part of the graph, okay? But the other one is a functional resource, the possibility to use genes to acquire new functions. Then we have to go a little bit more in detail. We cannot just have black boxes doing functions without giving names to the things we investigate. So the new era of the next generation sequencing, parasequencing and so on is allowing, and 454 metagenomics is allowing for the first time to have a full and complete spectrum of the viral diversity. And just to recall you some numbers, the total number of marine species discovered so far is 250,000 species. Overall on Earth, we have 1,700,000 species, okay? 250,000 species are censused now. In just a few grams of sediments, you can easily find more than 10,000 species, viral types, viral species, let's say. And overall, in 1 kilogram of sediments is easy to find 100,000 species. So in just 1 kilogram of sediments, just in terms of viruses, we can find half of the diversity for all other life forms inhabiting the oceans. Just to give an What are doing these viruses has been anticipated. They kill everything everywhere. But what we demonstrated in a paper that got the cover of Nature in 2008, and again, this was designed by Michael Tangalini, which is I think that I underline because I think that more and more science has to connect with the ability to show and to the graphic arts and to display things which are invisible, so we cannot, you know, just, it's not a picture of an elephant, okay, showing a virus killing prokaryotes, is that the deeper you go in the oceans and the higher the fraction of prokaryotes killed by viruses. So that in the abyssal, beneath 1,000 meter depth, more than 80% of the global microbial production is killed by viruses. So it's a huge engine guaranteeing the continuous functioning of these components, even in systems which are food limited. So in principle, they should run very slowly. So accelerating everything. So keep in mind this, because at the end, the big picture of this is that all life forms interact. I started saying that the Dumbo octopus, the Lamellibrachia, and others, the big clams, were integrating bacteria in terms of symbiosis. Then there are pathogenic bacteria. Then there are viruses killing the animals, viruses killing the protists, viruses killing the bacteria, and so on. Overall, all these interactions contribute to the ecosystem functioning. So the production of biomass, the production of energy, decomposition, and so on. But these components are not actually included in any ecological model dealing with biochemical cycles we have in the world. And we cannot neglect the components that rule the world. So we are facing a new frontier. The new technologies are allowing, you know, to do what was before impossible. Now we can go in the deep, we can manipulate. We are just back from a cruise with the Jamstack. We were putting experiments at 9,000 meter depth, you know, investigating what's going on in there if you inject certain substances, if you simulate the climate change occurring in the deep, and so on. We need, you know, sophisticated technologies, expensive technology. One ROV, remote operative vehicle like this, can cost up to 10 million euros. And every single day at sea with a cruise might cost 50,000 euros, 80,000 euros. So it's a kind of very expensive science, but a very also promising kind of investigation. Why? Because we now know that, for instance, by 2050, about half of the oil will be extracted at depth beneath 1,000 meters, so in the deep ocean. Because we know that huge amounts of gas hydrates are stored beneath 8,000 meter depth. And they are endangered due to climate change and to the warm up of the waters that can release the methane with climate feedbacks, because methane has an effect 20 times higher than carbon dioxide for altering the climate. And we are facing a new era in which more and more international companies are looking for using the polymetallic nodules and the rare metals they contain, or the pinnacles I showed by the hydrothermal vents. And there are also coming out papers saying how we can restore the deep, even though we still don't know much about these systems and how to restore it. So this is the last part of my talk. Is there any future for the oceans? Where we are going to, and how the microbes can contribute to the future of the ocean. A few words about what's going on there. We are discussing about global climate change. That means temperatures, but also in the ocean means decreasing pH, decreasing oxygen content. We are going to have, deoxygenated oceans in the future. Overfishing, habitat destruction, mining, for instance, drilling, pollution. This was the past. In the past, you used to go at sea and have a fish catch like this. Is that reasonable now? No. This is a fish catch. These are not professional, maybe fishermen. We are experiencing a measured decrease of the length of the animals, the biomass of the animals. This is a catch from a tonnara di Camogli in the Ligurian Sea. In the 60s and 70s, hammer sharks, white sharks, orcas were caught there. Now they are disappeared, and the abundance of all sharks in the Mediterranean has collapsed by more than 90 to 95%. This was a fish catch in California in the 50s. Look at the size of the catch. It was just a touristic, activity, a few hours at sea. This was the result in the 80s, and this in 2007. So this shows you, by just a picture, how it's changing the ocean in about 50, 60 years. So everything's changing. We are reducing everything. So the more we reduce the big to the small, and the more we will go to the ocean, which is populated just by microbes, small things. And we're coming back to the primordial ocean, when the life was born. It was just a soup of microbes and viruses. The collapse of fisheries is evident. There are projected trends that say that if we don't develop no-take zone areas, protected areas, we are going to fish up everything. And the erosion of food webs is such that we are changing the system for which we are not facing any more sharks or big tunas, but just jellyfish. And maybe sea turtles or moonfish, because they eat the jellyfish. But the system is changing. And we are destroying everything. This is the reef I showed at the beginning. This is after a trawling, a fish net passing by, catching fish there. And this is a permanent, because constructing this kind of reef takes maybe 1,000 years. And this is the result. Marine biodiversity loss is a major problem, not because we kill the animals, but because we kill the global functioning of life. In a study I published in 2008, we demonstrated that if you lose biodiversity, you lose functioning of the ecosystems exponentially. So you lose 20% of biodiversity, well, then the system works at 50%, at 20%. So there is a measure impact. It's not just a proportional decrease biodiversity versus functioning. There's a measured decrease, and that was called by the Science for Environmental Policy of the European Union. So we need to protect biodiversity to reduce the impact of ecosystem functioning, but still, already 1/3 of the species have collapsed, so decreased by 90% in terms of abundance. This is a paper that was published today by PLOS Biology, by Camilo Mora and myself and another group as a result of a workshop we ran a few months ago in the US, where for the first time we tried to see what happens, not just if we project the impact of temperature or oxygen, but we put together the impacts, decreasing oxygen, decreasing pH, increasing temperature, and so on. And we discovered that there is a synergistic effect, so is more than the sum of the impact of each variable. And the result, so, will be even harsher for life. So, concluding, what's the consequence of life for our life in terms of functional ecosystem? How we can link it back to the microbes, the effect of climates, and so on. This is a simple scheme of the interactions between the rising atmospheric CO2, the change in carbonate chemistry, that means decreasing pH in all oceans, the increasing sea surface temperature, which also increases the stratification of the warmer surface layer versus the deeper, cooler layer, and the interactions with the biological components. We know that phytoplankton and primary producers take up CO2, so they balance, they reduce, they use a large amount of CO2, which is stored in the ocean and which is also buffering the effect of CO2 emissions. But if they sink to the bottom and they are transforming hydrocarbons eventually by burying into the sediments, they are a permanent sink of CO2. If they are killed by viruses, they are transformed into dissolved compounds, they are respired, and they are coming back to the atmosphere. So the more we are able to shift between the system that goes from CO2 to phytoplankton to the deep, and the more we can buffer the climate change. But if we increase the viral impact on these components, thanks also to the increased activity of heterotrophic microbes, which respire the dissolved organic matter, we give back the CO2 to the atmosphere. So there is a very delicate and complicated interaction in this. That's not all. Let's look at the bottom of this. Once you produce the material and the material settles down to the deep sea floor, if you increase the temperature in the deep, you have that microbes, in principle, by increasing temperature, they increase the metabolism, they increase the activity, they respire more. So they will tend to use more organic carbon to release more organic carbon to the water and to the atmosphere. In that case, viruses killing the bacteria in the deep can shut down the process, can reduce the respiration in terms of, and can release the DOM, which is stored in the deep. So in that sense, an increased viral infection in deep sea components can also contribute potentially to the storage of organic carbon, and so the CO2 ultimately, in the deep ocean interior. Understanding which is the process prevailing in between the one giving back CO2 to the atmosphere or the other going down is difficult because it depends also on the effects of climate change on the different components. But it's not all. We know that the ocean can control the global climate, for instance, by producing cloud condensation nuclei. We know the story of the dimethyl sulfide released by phytoplankton that goes to the atmosphere and creates cloud condensation nuclei for creating the clouds. The clouds, protect, from irradiation, and reduce then the penetration of light, the production, and so on. But more recently, we also discovered that this process is not just mediated by the dimethyl sulfide, but is possibly even more dependent upon the release of very, very tiny organic particles of the size of viruses. which represent themselves condensation nuclei. So at the end, what is going to be realized now with the experiments we're running is that viruses, beyond the role of predators of the sea, they have also a role potential in the cloud formation because of the aerosol, along with the components of the organic components that are transferred from the ocean to the aerosol. They include also these tiny particles, that means the debris of the prokaryotes and the bacteria they kill, and the viruses themselves, that represent a huge amount of cloud condensation nuclei, which activate cloud formation and then regulate the climate of the biosphere. Again, giving light, shedding light to these processes indicates that a single component can influence many others in different ways. In one way, they represent maybe a physical substrate on which clouds are formed. In other ways, they contribute to the release of organic particles that they contribute to cloud formation. In other cases, they contribute or not to the sequestration of CO2. Whatever is the role of viruses, we need to investigate that further. We are just at the beginning of that. But it's clear that for many reasons, from the biotech perspective to the global climate regulation, they play a key role, not just in the oceans, but for the functioning of the global biosphere. We are now understood that the human society is influencing biodiversity, either directly or through the global climate change related to the climate-altering gases. We understand that all these components altering biodiversity can change ecosystem functions because they are tightly linked. Life is the one performing the processes we need to sustain other life, biomass production, carbon sequestration. All these processes are mediated by life in accordance to the physical constraints, chemical conditions surrounding, and so on. Functions are not just needed for life themselves, but are needed for us because they produce the ecosystem services we need. Without the oceans, we will have the double of CO2 concentration in the atmosphere because of the buffering effect. Recreational, water quality, waste control abatement, all these are systems given for free by a functioning of the ecosystem functioning, and in particular are given by oceans with benefits for human society. So we have to understand that human society has to do something to mitigate and adapt to this in order to not pay too high toll for that behavior. So we need to understand better life in this changing ocean, from the big to the small, from the waste to the microbes to the viruses. And we have to understand that there is a long way to go ahead. The target for protecting the marine environment is to protect at least 10% of the ocean surface. So far, we just realized 0.1% of the ocean surface, while inland, we already achieved the quota 10%. So this is a big engagement we have to take, all of us, to understand that the life of the ocean is also our life. The oceans produce one molecule of two that we breathe, So half of the oxygen produced on Earth is produced by the oceans, so they are vital for all of us. And I hope that the lecture I gave here contributed to your appreciation of this importance. Thank you very much for your attention.